Hi, welcome to our webinar today. This is brought to you by the ASQ Statistics Division. A couple of housekeeping notes. We ask if you have questions, if you type them into the Q&A or the chat function, and we will get to those at the end of the presentation. The presentation is being recorded, and you can access that later through the YouTube channel that we have for the Statistics Division. And if you have any issues, you can reach me through the chat function, and I will try to help you. We have Brooke Marshall here today to lead us on a topic on split plot designs. She is the head of CMC statistics at Moderna, leading a team of statisticians supporting process and analytical research and development, manufacturing control strategy, and stability modeling for mRNA vaccines and therapeutics. So very on-topic talk today. She has many years of CMC statistics experience in biopharmaceutical process and analytical development, as well as small molecule manufacturing and stability through her previous roles at GSK and Merck. She has statistical experience and design of experiments, response surface methodology, quality control, and regression modeling through her education at Virginia Tech, where she received her PhD in statistics. And I'm going to turn it over to Brooke.
These are really some of my favorite types. Oops, okay, oh, maybe I was muted. These are really some of my favorite types of designs because I think they allow a lot of flexibility in terms of how scientists and engineers can run their experiments. So I recommend them frequently. So just to give an overview of what we're going to talk about today, I'm going to talk a little bit about the historical uses of split plot designs and kind of try to set the stage for how these can be used in industrial settings. And then we'll walk through a pharmaceutical process development case study to really highlight the flexibility of these designs, and then talk about actually generating and modeling the split plot designs in JUMP. So just starting with the historical use of these designs, they were really kind of born out of agriculture experiments where we were doing crop studies, decades and decades ago, looking to really assess maybe like the yield of certain crops under different conditions. So in a lot of these crop studies, we'd be looking at factors such as irrigation method or fertilizer type to really determine what would give the best yield for crops. And so in these types of studies, something like irrigation, really an irrigation system needs to be applied to a very large area of land. And so we might apply that to what we call a whole plot or a large plot. And then other factors such as fertilizer type, which can be applied to a smaller area of land, can then be put within each whole plot as a subplot. So this is kind of where the split plot design name came from. It came from designing these agricultural experiments when you were literally splitting plots of land into different sections. So in this case, our whole plot factor is our irrigation method because it's applied to a large plot of land, and our fertilizer type is then replicated within each whole plot and randomized. So what this really does is it gives us different size experimental units for our different factors of irrigation method and fertilizer type. And it's because the irrigation method is over a different area of land than the fertilizer type. So this really impacts how we model and look at the results. So I'm going to show you just a partial ANOVA table. So anyone who's familiar with doing any sort of analysis of variance, I hope this looks somewhat familiar. This is a partial ANOVA table showing all the terms that you would fit for this split plot design. And it's very similar to what you would see for a two-way ANOVA. So we have our factor A of irrigation method, we have our factor B of fertilizer type, and we have our AB interaction. But there are some differences here, the main one being that we have this additional error term for the whole plot, in addition to our residual error, which is really the split plot error. So the reason that we have this is because we have those experimental units of different size where the irrigation method is essentially being applied over a larger area of land than the fertilizer. So what we have to do then is estimate different variance terms in order to do the appropriate test for these different factors. So in order to test irrigation method to see if there's differences between those, we really need to test against the whole plot error term. And for the split plot terms, which are the fertilizer type and the interaction, we would use the split plot error as the denominator in our test statistic for that. So these models are typically fit with a mixed model, and we do that so that we can estimate this whole plot error term and the split plot error term, which would be variance components in our mixed model, and then the rest of the model terms would be modeled as fixed effects. So the other part of this ANOVA table that's important are the degrees of freedom, and I don't want to get bogged down in what they are and how to calculate them, but just say that they're really giving us an idea of how much information we have to estimate each term in the model. So this would be really similar to sample size if you're thinking about how much information you have that goes into some sort of estimate. And it works the same way. So if we have lower degrees of freedom, it means that we have less power to test one of our factors or interaction terms in the model. So the main idea here, or the main thing to show, is that our whole plot error has 1 degree of freedom, where our split plot error has 6 degrees of freedom. So what this means is that we have a lot less power to detect differences in our irrigation method compared to our fertilizer type. So we actually have, I guess, a little bit of a trade-off here in terms of power when we actually implement these designs. Because we don't randomize the whole plot as frequently as we randomize the split plot, we end up having less power because we end up having less replicates. Okay, so just moving on to kind of elaborate a little bit more on these historical examples. You can further split these plots into more sections. So say, for example, we wanted to study irrigation method and fertilizer type, but we also then wanted to add another factor like crop variety to see what its impact is on crop yield. So you could then split the subplots a second time and plant different varieties of a crop. So in this case, we have 2 irrigation methods that you see are replicated twice, because we have irrigation method 1 twice and irrigation method 2 twice. And then we have the fertilizer type that is 1 through 4. So there's 4 different types randomized across each of the irrigation method whole plots. And then within each one of those subplots, we have randomized every single variety of 4 types of crop. So in this case, we actually have experimental units of 3 sizes instead of just 2 like we did in the split plot design that I just showed. So the ANOVA table for this looks a little bit different. You'll see that the top half of this ANOVA table looks very similar. It's basically exactly the same as for the split plot design. We have our irrigation method, factor A, our fertilizer type, factor B, and the interaction, along with the whole plot and split plot error. But now we have a whole other section for our split plot. So we have our crop variety and all the interactions associated with that. And then we have our split plot error term. So in this case, because we have 3 different sizes of experimental units, we also have 3 different error terms that we need to test against when we're looking for differences in these factors. But the same thing applies in terms of the degrees of freedom. So the lower the degrees of freedom, the less power we have to test the factor or interaction. So in this case, with the whole plot error, we still just have 1 degree of freedom. But you'll see in the split plot, we have 24. So we have pretty high power to detect differences between crop varieties compared to fertilizer and irrigation methods. All right, so just one more thing about using this historical design as an example before we move on to the industrial applications. I just want to bring this point to the forefront because I think it's kind of a pitfall that I see a lot when people are trying to implement these designs. So one of the biggest things is that you actually really truly have to replicate those whole plots, because if you don't, then what happens is your degrees of freedom go to 0 for all of your error terms. And if that's the case, then you can't estimate your error and you won't be able to test any differences between your factors like irrigation and fertilizer type or your interaction. So you need at least 2 whole plot replicates in your design in order to do your statistical test of significance. And of course, the more whole plot replicates you have, the higher power that you'll have for your whole plot factors. All right, so I'm going to move on to talking about these designs more in an industrial setting. So these split plot designs are really commonly used in scientific studies when we have factors that we say are hard to change. So a hard to change factor is one that really can't be randomized for every experimental run when you're in the lab, for example. And a very common hard to change factor is temperature. And it's actually going to be in the case study that we show. So just to give some idea of how these can be applied, one area is in analytical development. So you can imagine if you're running, say, some assays on a plate, and you have multiple assay runs from different samples that you've put on the same plate, but you may want to incubate that one plate at one temperature because you can't really split the wells from the plate. So you just put it in the incubator. Now what happens is your temperature becomes a whole plot factor because it's essentially not randomized across all the runs on the plate. So in that case, because of the lack of randomization, that becomes a whole plot factor, and this would need to be analyzed as a split plot design. So then another example is in process development, where you might want to study different process parameters across multiple steps of a process. And in this case, you can really only randomize the process parameters that are at the corresponding step. So what happens here is that your randomization ends up being restricted for your upstream factors at each downstream process step. And this process development type example is what we're really going to get into in the case study to kind of show how this would really work in practice. So this is actually something I worked on at Merck many years ago when I was in small molecules, and we were making tablets with 2 actives. So just to kind of describe this process, we start with making what's called a spray-dried intermediate. And the spray-dried intermediate is basically created by taking your active ingredients along with the excipients and dissolving them in some sort of solvent and then putting them in a drying chamber. And so at the end of that, what you end up with is a powder that contains your active ingredient. And then the next step of the process, because you have this powder and you eventually want to form it into tablets, you actually have to make it into granules first. So we do that in a granulation step by rolling that powder through a roller compactor. And then finally, we take the granulation and we press it into tablets. So really what the goal of this experiment was to determine how the dissolution of the active ingredients was impacted by the process variables at each step. So in the first step, where we were creating the spray-dried intermediate, we had 2 active ingredients, and they were each, the spray-dried intermediate was created for each active separately. And we wanted to look at how the temperature of the drying chamber might impact dissolution. So we had 2 different temperatures here, one for each active ingredient. Then in the granulation step, where we're running it through the roller compactor, we just wanted to study one factor, which was the roller compaction force. And then finally, when pressing the granulation into tablets, we wanted to look at how hardness and moisture impact the dissolution. So basically how this works is that at each step, we start with whole plots at the spray-dried intermediate, and then the next step becomes the split plot, and the final step of this process becomes the split plot. So I'm going to describe how this works in terms of the split plot design. So at our first process step, let's assume, and this was the case for this particular example, we had 2 levels of temperature for each of our whole plot factors. So remember, these are just the temperatures that of the chamber to make our spray-dried intermediates for the active ingredient 1 and active ingredient 2. So if we have each temperature at a high and low level, then essentially what we have is a 2 squared or 2 to the 2 factorial structure in the factors for the whole plot. And that gives a total of 4 combinations for 1 replicate of the whole plot. So you'll see on this top row, you essentially have every combination, low, low, high, 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 low, and low, high for the 2 temperatures in the drying chambers. And then we want to replicate that, these 4 level combinations, again, because we need to have at least 1 replicate of our whole plot. So what we ended up with at the end of this step is we started with, our raw materials and created essentially 8 batches of spray-dried intermediate bulk that each contained 2 active ingredients that were made from drying at 2 different temperatures. So if we think about this, now we have these kind of 8 batches that we're moving into the next process step. So the second process step is to make the granulation. So in this case, we're only looking at roller compaction force as our only factor, and we were going to do this also at 2 levels, a high and a low level. So essentially now you can think about, I have these 8 batches of material that were created in the last process step, and now I'm going to split each one of those into 2, and I'm going to make granulation by running half of that batch through a roller compactor at low force and half of it through high force, and I'm going to randomize that for each batch. So now at the end of this, I end up with 16 batches of granulation to press into tablets at the next step. So now we had 2 factors we were looking at studying on the third process step. So these were hardness and moisture. So essentially now we want to take each of those 16 batches that we got from the last process step and split them into 4 because we want to run, or we want to press them into tablets at different hardness and moisture settings. So again, we just chose 2 levels for each one of these factors, a high and a low. So you'll see in this diagram, essentially for each of the 16 batches from the previous step, we split that into 4 to get our hardness and moisture at low, low, high, 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 low, and low, high. So this again has that 2 by 2 factorial structure that we had in the whole plot. We now have that also in the split plot. So what this ends up giving us at the end is a 64-run design where essentially at the end you can think of it as having 64 batches of tablets that we can now assess the dilution on and get our response. So let's just quickly look at the ANOVA table for this setup to see what this looks like. It's much more complicated than the historical examples that I showed, and this mainly comes from the fact that we're looking at now 5 factors, and we're looking at a lot of the interactions between those and the fact that they have factorial structure in the whole plot and the split plot. So just to take a look at the whole plot terms, you can see that we have the main effect for our spray-dried intermediate temperature for active 1. We have the main effect for the spray-dried intermediate for active 2, and we have the interaction between those 2. So those are all terms that are associated with the whole plot. Then when we move to the second process step where we're doing the roller compaction, we have the main effect for that, as well as the interactions between the roller compaction and everything else that was in the whole plot. So those are all those interactions here. Then in the split plot, we have moisture and hardness, as well as the moisture and hardness two-way interaction. And then again, we cross all of those with everything in the split plot above and the whole plot above that. So we have a ton of different interaction terms that we can estimate now with this 64-run design. We actually end up with 27 main effects in interaction terms because of these 5 factors, and we have to test them all against the appropriate error term. So in this case, it's the same as before. We have really high power for our split plot terms and maybe not as much for our whole plot and split plot terms. But the next question is really, do we need to do all 64 of those runs? And the answer is no, because of 2 things. One is that we typically don't need to estimate higher order interaction terms because they typically don't truly exist. So we can think about actually fitting a smaller model than we needed to, than we would be able to fit with the 64 runs, because we just don't really need to do that. So here I've kind of highlighted all of the 3-way interactions, 4-way interactions, and even the 5-way interaction that we get from this design, because we can really probably drop those from the model. And it means we probably don't then need as many runs because we don't have to estimate these terms. The other thing is that we have high power in the split plot because we have those 24 degrees of freedom. So we don't really need these extra runs to increase our power in the split plot because we have plenty already. So what we can do then is actually start removing some of the runs from this design. And we can do that because of the factorial structure that's in the split plot. So because of that structure between the hardness and moisture, we don't really need to do every single combination after all of the spray-dried intermediate is made and after it goes through the roller compaction to get granulated. So we can take what's called a 1/2 fraction of a design, And we choose that half fraction based on aliasing structure or confounding of certain terms. So we know we don't need to estimate our four-way interactions and four-way interactions and three-way interactions. So what we can do is we can confound our four-way interactions with the main effects, and we can confound our three-way interactions with the two-way interactions. And by doing that, we can actually remove 32 of these runs and end up with a 32-run design. And this is still what we call a resolution 5 design, where we can estimate all main effects and two-way interactions without any confounding of the terms. So what we ended up with, and this was the experiment that was run, the final design that was selected was just doing basically 4 runs for each whole plot all the way through the process. So this was really helpful to reduce the number of runs because, of course, we didn't have to use as much raw material. But then also, it really saved the lab because instead of running 64 dilution tests, they really only had to run 32. So a lot of times this can save a lot of time and money in trying to reduce these designs at the split plot level. So the way that they actually performed the experiment, because they couldn't run all of these whole plots on the same day and they needed the replicates, they actually ran one whole plot replicate on one day, and they did another whole plot replicate on another day. So the reason that I want to bring this up is because it means that the whole plots have kind of a certain structure. And the structure that they have is a randomized complete block design structure, or what we call RCBD. So the whole plots are then essentially arranged in a randomized complete block where one replicate of the whole plots is in one block and the second replicate is in the second block because they were run at different times. So all of the ANOVA tables that we looked at so far, they have this top line that says whole plot replicates. Essentially what that term is doing is adjusting for the fact that you have your whole plot replicates in blocks. So this is really an important term to add when you are running your whole plot reps in blocks. And typically, when you're doing these types of experiments, it is impossible to do all your runs at one time. So you typically do have to block your runs or every whole plot rep into a block. But in some situations, you may not need to do that, and you can actually do what's called a completely randomized design whole plot structure. So this is just kind of backtracking to the beginning where we were creating the whole plots for this type of design. So we could have actually assigned the levels for the two drying temperatures a little bit differently. We could have done it in a completely randomized way, where every combination had an equal chance of being in any whole plot. So then in that case, you can see in this particular example, you could have had whole plot 1 and whole plot 3 be at the low level, or in this example, a whole plot 6 and whole plot 8 are at the high-low level. So these are no longer in a randomized complete block design structure. They're now in a completely randomized structure. And I just want to point this out because it does change how you fit your model. So where we had that term for the blocks of the whole plots, we now essentially lose that term because we don't need it anymore. And those degrees of freedom go back into the whole plot error, giving us a little bit more power to test the whole plot model terms. But like I said before, it's usually unlikely that you could even do this type of design because it's usually impossible to do all your runs at one time. So the randomized complete block design structure is the most common for these types of experiments. And just to, I guess, make another point, the whole plots can really be in other structures too. Like if you're familiar with Latin squares, they can be in a Latin square structure. So they can really be in any kind of structure that you can create from other types of designs. All right, so that's all I'm going to say about the case study. And now I just want to talk a little bit about how to fit these models in JMP. And we'll just use the case study, though, as an example. So basically, hopefully, people have access to JMP and will be able to create these designs there. I think it's the easiest software to use for these designs. You can go into the custom design platform in JMP and essentially just include any response variables that you're going to measure. In this case, we were really looking at dissolution. And then you can enter all of your factors. So here we have our chamber temperatures for the spray dry intermediates. We have our roller compaction force factor from the second process step, and hardness and moisture from the third process step. So the way that you tell JMP that these are whole plot or split plot or split plot terms or factors is really by telling it is how hard to change it is. So here, for the whole plot factors, I've said that they're very hard to change. That means they're going to be randomized over the very least of anything else in the design. Then your split plot, which in this case was just one variable roller compaction force, I say it's hard to change. So then JMP knows that that is going to still have restricted randomization, but it's not as hard to change as the chamber temperatures. And then we select easy to change for our split plot factors because they can be randomized the easiest within each of the split plots. All right, so once we've basically told JMP how hard to change our factors are, we need to tell JMP what model we're going to fit so that it can generate the design, the best design for the model. So automatically JMP is going to give you a model that includes the intercept and all of the factors, the main effects. But then we wanted to really look also at two-way interactions. So you can just click the interactions button in JMP and select two-way, and it will give you all of the two-way interactions. So this is the model that we were choosing to fit. The next thing that you have to specify in JMP are the number of whole plots, the number of subplots, and the number of runs that you're going to do in total. So JMP will automatically give you some default number of whole plots, subplots, and runs. So in this case, the JMP default gave us 6 whole plots, 12 subplots, and 36 runs by default. So if I clicked Make Design from this, it wouldn't give me the exact design we created for this particular experiment. And they select 6 whole plots, which actually means that there's no way that you would have a completely balanced design with your replicate, with having every single combination of the 2 spray dry temperatures in the whole plots twice. So essentially, to create the balanced design and a half fraction that we were looking for, we actually just decided to change these numbers. So to make it balanced, we changed the 6 to 8 and the number of subplots from 12 to 16. And then to get that half fraction, we actually specified the number of runs that we wanted at 32. So then JMP generated a design, and this design is probably not exactly the same as the one I showed on the previous slides, but it's definitely comparable. You can see that for your whole plots, so you have a whole plot column here, your first whole plot, there's no randomization happening for either of the factors in the whole plot. But then you do have some randomization as you go down to the roller compactor step and then the tablet pressing step. So that's really how this works. And so the subplots, you can see the first subplot, there's no randomization between roller compaction force for each of the subplots. So JUMP has really created the same type of design that we generated to run this experiment. And once you've generated the design, you can also look at the power. And this is really important to assess. So I think in relative terms, though. So unless you have some good idea from historical data what your standard deviation is or your root mean squared error, which you could enter here in JUMP, and you have some sort of idea of what your coefficient estimates are in your model, JUMP just by default puts a 1 in all of these boxes. So the power numbers that it's giving are not necessarily the power that we're going to have, since we're not sure what these coefficient estimates are or what our standard deviation is. But it's still important to look at this because it tells you relatively how much power you have for your different model terms. So in this case, everything that's with a red box around it are things from the whole plot. So these are the chamber temperature for active 1, active 2, and the interaction. And you can see that the power for those is 0.374, which is the lowest power of anything listed here. And that's really because these are the whole plot terms. We know we have less power because we know we've replicated them the least number of times. Then at the second process step, or the split plot part of the design, we have the roller compaction force, and we have that crossed with both main effects from the whole plot. And you'll see that then the power increases for the split plot to 0.743 for all of these, which is better, but it's still not the best. We get the best power from the rest of the terms, which are from the split plot, because we've randomized hardness and moisture the most. So this really kind of shows the trade-offs that you have with power when you can't completely randomize your design and you have some sort of restricted randomization like you do with a split plot. So the great thing about JUMP is once you've specified all of this, JUMP will generate the appropriate model in its fit model platform, so you don't even have to worry about selecting the appropriate model. JUMP does that for you. All you have to do is really enter your response variable, and then all the model terms are populated for you. And I just want to highlight that at the top of the list are always going to be your whole plot and subplot or split plot error terms that it's estimating with the mixed model. And JUMP knows that these are going to be used to test the appropriate terms in the model. So that's all I'm going to say about JUMP. And before I think we open it up for questions, I just want to leave you with this one quote that I think about a lot, which is that all industrial experiments are split plot experiments. It's been my experience, and I think for anyone working in a lab or on process development, you can typically find something that you're, some factor in your design or something that you're doing that you really can't randomize. In that case, you really should be considering using a split plot design and model to analyze your data.
Great. I just want to remind everybody there's a chance for questions. If you have any questions. Go ahead and type those in. We will be posting the presentation in the YouTube section of our YouTube channel. Brooke, do you have any books that you recommend on this topic?
Oh yeah, I meant to put some references, but I didn't. I personally don't have any books that I've gone to. I mean, I've learned about this in my design class. I think there's a lot of great articles out there, like Brad Jones and Chris Nachtsheim have a great paper about split plot experiments and why and when and how to use them. But I think it's a good idea to just do a Google search for articles as opposed to trying to look something up in a textbook on these.
Great. Does anybody else have any questions? Doesn't look like it. It was a very clear presentation. I really enjoyed it. Thanks, Brooke.
Okay, thank you.
I will be sending out the email for the RUs for your continuing education, and then within a couple days we'll have the video posted to our YouTube channel. So thanks everyone for attending, and hope to see you in a future webinar. Thank you.
Thanks.